So friends, let's come to the last part of the anatomy of the ear, that is the neural pathway of sound. Now how this sound goes from the inner ear, that means the cochlea, to the brain. So to learn this neural pathway, you see, we have already seen this diagram. This is a diagram of organ of Corti, and as I showed you, from this organ of Corti starts the cochlear nerve. Okay, cochlear nerve is the eighth nerve, vestibulocochlear nerve. So it has two parts, the vestibular nerve and the cochlear nerve. So this is the part of the eighth cranial nerve, and this takes the stimulus from the inner ear, sensory organ of Corti, to the brain. Now some people, they ask, from where does this cochlear nerve starts? So you see, although the inner hair cells, they give 90% of the stimulus. Okay, so that means 90% of the fibers, they are at the inner hair cells. But you see, the 5% stimulus is also by the outer hair cells. Moreover, now we believe that it is like a feedback stimulus to the outer hair cells to regulate the sound. So if somebody asks you from where the eighth nerve fibers are starting, look in this diagram and answer outer hair cells. Okay, so from the outer hair cells, not just the inner hair cells, from the outer hair cells, these fibers are starting and they go to the eighth nerve. Now to remember this pathway, we can learn a mnemonic, and the mnemonic is E. coli ma. Okay, E. coli ma. Right? Let's look at this diagram. Now we know the sound goes from the cochlea to the brain. Okay, so let's start from the right cochlea, and the mnemonic is E. coli ma. E over here stands for eighth nerve, the vestibulocochlear nerve. Okay, from here it goes to C. C is cochlear nucleus. Right? So E. coli ma. Okay, so E is the nerve, eighth nerve. Right? C is the cochlear nucleus. Now remember, the cochlea is a spiral structure. So therefore its nucleus is also spiral. So therefore nucleus is also known as spiral ganglion. Right? So cochlear nucleus is also known as spiral ganglion. Now where is this cochlear nucleus? This cochlear nucleus is situated in the lateral recess of fourth ventricle. Okay, lateral recess of fourth ventricle. Right? Lateral recess of fourth ventricle in the brain stem. Okay, so that means it is in the brain stem we have the cochlear nucleus, also known as spiral ganglion. Now from here the sound goes to O. What is O? O is superior olivary complex. Okay, O is superior olivary complex. Now remember, O is also for opposite. O is also for opposite. So you see, now the sound has gone to the contralateral side. Right? The sound has gone to the contralateral side. There is a crossover of sound. So this question is asked that the crossover of sound takes place at, your answer is superior olivary complex. Remember, O for opposite. This superior olivary complex lies in the trapezoid body. So in the brainstem we have the trapezoid body, in that lies the superior olivary complex. So sometimes the trapezoid body is also asked in the question. So remember, this is involved in the auditory neural pathway. Okay, so from O the sound goes to L. L stands for lateral lemniscus. L also stands for largest. So that largest L, why I am saying L for largest, that I will explain later. Just remember that L stands for lateral lemniscus. Okay, from here the sound goes to I. I is inferior colliculus. So now you see cochlear nucleus, superior olivary complex, lateral lemniscus, inferior colliculus. These are all part of your brain stem. Okay, so that means the coli that I have just shown you over here, cochlear nucleus, superior olivary complex, lateral lemniscus, and inferior colliculus. This is all part of brainstem. And what is ma? The ma is cerebrum. Okay, so you see E. coli ma. The mnemonic not only tells you the pathway in the sequence order, it also tells you which part of the pathway it is, brainstem, or it is nerve, or it is cerebrum. So what is ma? M stands for medial geniculate body, and A for auditory cortex, Brodmann's area 41. Right? Now you see the sound started from the right side, and the majority of sound has gone to the left auditory cortex. But if you notice, some amount of sound has also gone to the right auditory cortex. So that means one side cochlea, if it receives the sound, it will stimulate both the side auditory cortex. Okay, so remember, sound is a global signal. Global signal means it stimulates both the sides of your brain. Okay, the right as well as the left auditory cortex. Right? Okay, so now we have seen the auditory pathway. Let's solve this question. Arrange the sequence of auditory pathway from peripheral to central. This question was asked in the AIIMS May 2019 exam. So now the simplest way is, we already know the mnemonic. The mnemonic is E. coli ma. Okay, so now how we solve this question? We read the choices. Inferior colliculus. Okay, choice A, inferior colliculus. So I mark it over here. Okay, A choice. And then we have the cochlear nucleus, so I mark the B choice over here, below the cochlear nucleus. Auditory cortex, C choice, so I mark it over here. And medial geniculate body, so that is over here. So now the pathway is B to A to D to C, right? That is our answer. So very simple, very quick, you can arrange in this way. Just write the mnemonic first and start putting the options below the mnemonic, right? Okay, now let's come to BERA. What is BERA? Brainstem evoked response audiometry, also known as ABR, auditory brainstem response. You see, we have studied the neural pathway of sound. What is that neural pathway of sound? We have studied all these different levels. What is happening at these levels? Each of these places, E. coli ma, each of these places, what is happening? One neuron ends and the other neuron starts. That means the electrical signal is passed from one neuron to the next neuron. Right? Okay. So when the electrical signal is passed, some electrical signal is lost. It is lost outside. It leaks out. Okay. And it produces an electrical wave in the brain. Now, if we study these electrical waves, we call it as BERA, brainstem evoked response audiometry. So what is this BERA? This is the study of electrical waves. Okay, so this is the study of electrical waves produced along the neural pathway of sound. Okay, produced along neural pathway of sound. So neural pathway of sound, what is this neural pathway mnemonic? The neural pathway mnemonic is E. coli ma. Right? Okay. Now, this study is only of the brainstem. So you see, till the brainstem we have to study. So we study the nerve, we study the brainstem, we are not studying the cerebrum. So you can remove the ma. Okay. And the mnemonic for BERA, therefore, is double E. coli. Why? I will tell you in some time. It is not E. coli. E. coli ma was the mnemonic for the neural pathway. The BERA mnemonic is double E. coli. Right? Okay. So you see double E. coli. Now the wave 1 of BERA is produced by distal part of 8th nerve. So 8th nerve is a long nerve from the cochlea to the brainstem. So it has two waves. Wave 1 is produced by distal part of 8th nerve. Wave 2 is produced by proximal part of 8th nerve. That is why we have two E's over here. Now, what do we mean by distal or proximal? You see, distal means away from brain. Proximal means near the brain. Whenever we talk about the nerve, the distance is always measured from the brain. It does not matter whether it is sensory or motor, afferent or efferent, whether it is taking signal from the brain or bringing a signal towards the brain. Distance of nerve is always measured from the brain, right? Okay, so distal part of eighth nerve, that means it is away from brain near the cochlea. So right at the start of the eighth nerve near the cochlea, the wave 1 is given. Wave 2 is away from the cochlea, near the brain stem. So the eighth nerve gives two waves. So that is why we have double E. And then we have the coli. The wave 3 is given by cochlear nucleus. Wave 4, O is superior olivary complex. Remember, O is also for opposite. Okay, over O happens the crossover of sound. Right? Wave 5 is L is lateral lemniscus. Wave 6 is given by I, inferior colliculus. Okay, you can even say wave 6 and sometimes wave 7, they are given by inferior colliculus. So this is your Bera. Let's look at the waves of the Bera. Okay, now I said L, remember L is the largest. Okay, so wave 5 is produced by L. L is for lateral lemniscus and L is also the largest. So wave 5 is the largest wave, or you can say the tallest wave. Okay, wave 5 is the largest wave or the tallest wave. Now this wave 5, you can see over here, it is produced at 5.5 milliseconds after the stimulus. 5.5 milliseconds after the stimulus. You see 0 over here is the stimulus when you have given a sound stimulus, and then different waves are being produced. So 5 is produced at 5.5 milliseconds after the stimulus. It is a stable wave. Similarly, wave 3 you can see over here, it is produced at 3.5 milliseconds after stimulus. Wave 1 is being produced at 1.5 milliseconds after the stimulus. So wave 1, 3 and 5. These three waves are stable waves. You remember the prime numbers in mathematics? 1, 3, 5 are prime numbers. So similarly 1, 3, 5 are stable waves. And you see the fifth is the most stable wave. Right? The fifth wave, the largest, the tallest wave, this is also the most stable wave. Okay, it is produced at 5.5 milliseconds after the stimulus. So largest, tallest, most stable, therefore this is the most significant wave also for us. Most significant wave for us. Now you see the distance between wave 1 and wave 3, this is how much? 2 milliseconds. Okay, distance between wave 3 and 5, this is again how much? This is again 2 milliseconds. Okay, so total distance between 1 and 5 is 4 milliseconds. This is known as wave 1 to 5 interpeak latency. Okay, and how much is the normal interpeak latency? That is 4 milliseconds. Interpeak means the difference between the peaks of wave 1 and 5. So interpeak latency. Latency means time delay. So if the wave 1 comes and then the wave 5 comes after 4 milliseconds of wave 1. Alright, so that is known as wave 1 to 5 interpeak latency. That is 4 milliseconds, 4.0 milliseconds. Now let's do some questions on Bera. Right? Okay, suppose you do a Bera. You do a Bera for a patient, you give a sound stimulus. Okay, you give a sound stimulus and you see that there is no wave. There is no wave at all. Okay, or the examiner says wave 1 absent. So wave 1 absent means all the remaining waves will also be absent. Okay, so no wave or wave 1 absent. So that means the sound has not been given by the cochlea to the eighth nerve. Okay, the sound has not been given by cochlea to eighth nerve. That means patient has problem in the cochlea. Okay, so that we can call this first situation, we can call as cochlear deafness. right? Or you can also call it as sensory hearing loss. Okay, sensory hearing loss, cochlear deafness or sensory hearing loss, right? Now let's say there is a situation, wave 1 present, all right? Okay, so wave 1 is present, but the remaining waves are absent. Okay, so remaining wave, which is the most significant wave, wave 5. So it says wave 5 is absent. Now wave 1 present means the cochlea is all right, it has given sound to the nerve, but the nerve is damaged. That is why the remaining wave, the wave 5, is absent. Okay, so this means the problem is in the neural pathway. This is neural hearing loss. Okay, it is beyond cochlea. All right, so it is also known as retrocochlear deafness or retrocochlear hearing loss. Right, so this is neural hearing loss or retrocochlear hearing loss. Right, okay, now let's say there is a tumor on the 8th nerve. The tumor has not completely destroyed the nerve, but because of the tumor, the nerve is stretched. Now, when the nerve is stretched, what will happen? What will happen? The wave 1 will be present. Okay, of course, that is near the cochlea, so that is before the tumor, wave 1 is present. Now, the wave 5 is also present, but the wave 5 is delayed. It comes after more time. Okay, so wave 5 is delayed, right? How much delayed is significant? Now, if the wave 1 to 5 interpeak latency, wave 1 to 5 interpeak latency is 4.4 milliseconds or more than that. It is 4 was normal, 4.0 was normal. If it is 4.4 milliseconds or it is more than that, that means it is abnormal. That means there is a problem in the retrocochlear pathway, in the neural pathway. So once again, we will call this as retrocochlear hearing loss or neural hearing loss, right? So 4.4 milliseconds or more than that will be known as retrocochlear or neural hearing loss. All right, okay, so this is how we solve questions of BERA. Okay, in your exam, suppose you get a question of the BERA, and in that question, suppose you are shown waves like this. Suppose this question is asked. In fact, this question was asked few years back. Now, what kind of hearing loss is this? Now, you see, BERA is not like ECG. In the ECG, when the stop heart's beating, it's a flat line. Now, over here, suppose the neural pathway is dead. There are other pathways, other neural pathways in the brainstem which are still working. So, some electrical activity is going on. Some waves are being produced. The patient is not brain dead, right? So, some electrical activity is seen. You can see some waves, but there is no recognizable neural wave for sound. Okay, so the examiner may say there is no wave, or examiner may say there is no recognizable wave. Okay, that means the same thing. And he may draw this diagram also like this. So that also means no recognizable wave, and that means it's a cochlear hearing loss because even wave 1 is absent. Now, if he says wave 1 present, remaining waves are not recognizable, it's a retrocochlear hearing loss. Okay, now let's say he draws the wave 1 over here. Now, let's say he draws the wave 1 at its position, and then he delays this wave 5 like this. Okay, so he produces wave 5 like this. Now, this is a interpeak latency has increased between 1 and 5, so that will be a retrocochlear hearing loss, most likely a tumor which is pressing the nerve and stretching the nerve. Okay, so just remember, this is how we solve the questions for the BERA. All right, okay, now this BERA is the confirmatory investigation for neonatal deafness. So what was the screening investigation? The screening investigation was OAE, autoacoustic emissions. BERA is the confirmatory test. So all those kids in which we suspect there is some problem in the OAE, then we do further testing by BERA. BERA is a confirmatory test. Remember, sensitive test, the most sensitive test is used for screening. Now, BERA is a very specific test. So, specific test is used for confirmation. Okay, so BERA is a specific test. Also, this test takes a lot of time. It takes around 30 minutes to 45 minutes. So, it is unfit to be a screening test. Right? So, therefore, this test is done in only those kids in which you suspect that there is a problem. You don't do it in all the kids. Right? So, remember this question, findings of BERA in vestibular schwannoma. So, we now know vestibular schwannoma is an 8th nerve tumor. Okay, so now what will happen? The 8th nerve is stretched. So, the wave 1 to 5 interpeak latency will be increased. So, we have already discussed that increased latency in wave 1 to 5. Decrease is wrong. 6 to 7 again wrong. Okay, decreased again wrong. So, now answer is increased latency in wave 1 to 5. Okay, and as I was telling you, BERA is the confirmatory test for neonatal deafness. Now, sometimes a question is asked, which is the best test for neonatal deafness? So now whenever the word best is used, we always go for the specific test. Best test for neonatal deafness. So now this question is asking about one neonate. It is not asking about a population of neonates. Okay, so best test for neonatal deafness. Answer is BERA. Okay, answer is again BERA. Somebody modifies this question and asks you best test for screening of neonatal deafness. Now he has added the word screening over here, therefore the answer will become OAE because he is specifying best test for screening. Okay, so screening of neonates is done by the OAE. See, this is how you can modify the language of the question. Otherwise, straightforward question, best test for neonatal deafness, it is asked about the specific test. Therefore, the answer is BERA. Alright, now I have specified this because some books have given controversial answers over here. So, this is what you have to remember and mark in your exam. The test for screening is OAE, test for confirmation, specific test is BERA, and best test is neonatal deafness. Okay, is again BERA. Now, somebody tries to modify and confuse you, best test for screening of neonates. So, yes, I again added the word screening. So, therefore, now he specified we are doing screening. That means a large population, it will be OAE. Otherwise, best test, BERA. Right? Okay. So, that was about the neural pathway of sound.